Hi, I'm William Foster, and today I'm going to talk you through our paper which we recently published in Cell Reports Medicine, entitled Tfh cells and the Germinal Center are required for memory B cell formation and humoral immunity after ChAdOx1 and COVID-19 vaccination. So I'm going to talk you through the data that we have in the paper and give a bit of background about the science and also about the techniques that we used. So to start the paper, we introduced the model, and this is to intramuscularly vaccinate mice with either ChAdOx1 and COVID-19. This is the Oxford AstraZeneca COVID-19 vaccine, which has been used worldwide, and a comparator, which is ChAdOx1 over. So this is the same viral backbone that the vaccine uses, but instead of introducing COVID-19 spike protein, it introduces over. And then we look at various time points downstream of this to analyze the resultant lymphocyte populations. In supplementary 1b, we show the pre-gating that we use to define our B cells. So these are live, single lymphocyte, B220, CD19 positive cells. And continuing on with the supplementary figure 1, we look at germinal center B cells in C, and we define these using BCL6 and Ki67 double expression for both the ChAdOx1 and COVID-19 immunized and ChAdOx1 over immunized mice. And in D, you can see in blue the nCoV-19 and in green the over immunized mice, the number and frequency of germinal center B cells. They're detectable at day 7 and their frequency peaks at day 14. But we see few significant differences in the number of cells here. So nCoV-19 and over ChAdOx1 vaccines both generate germinal center B cells. In E and F, we begin to look at antigen-specific B cells, and here we are looking at RBD-specific. This is the receptor binding domain, a key domain of the COVID-19 spike protein. And so you can see that in the nCoV-19 immunized mice, these RBD-specific cells are there, but in the over-immunized mice, these cells are not there, and this is what we would expect. So in the resultant graphs in F, you can see in blue the ChAdOx1 and nCoV-19 immunized mice have a large number of RBD-specific germinal center B cells, and the green line at the bottom, there are no RBD-specific B cells or GC B cells generated by the ChAdOx1 over immunized mice. Then we move on to the main figure 1, and here we look at class switched IgD negative B cells in A. So these are cells which have become activated and changed away from being IgD positive, and then we gate on RBD specific cells after this. And much like the supplementary figure as you see in B, the ChAdOx1 and nCoV-19 immunized mice generate class-switched RBD-specific B cells, and the ChAdOx1 over-immunized mice do not. Continuing on, we then took our IgD-negative RBD-specific B cells from 4 different time points, combined them together and ran a t-SNE clustering analysis, which is shown in C. In D, we have the heat map, which describes the various proteins which we detected by flow cytometry, which were used to generate the t-SNE. And from the t-SNE, we're able to describe 6 clusters of cells, and we can show how lowly or highly expressed each protein is on average on the cluster of cells. And from this, we can get an idea of the identity of each cluster of cells. For example, the largest cluster is highly expressed of BCL6 and Ki67. These are therefore the germinal center B cells, and so we then went and did conventional flow cytometry gating to find these various clusters shown in D. So we have the Ki67 and BCL6 double expressing germinal center B cells. Then we have the non-germinal center component. In this, we can find various clusters. For example, CD44 expressing, CD11c expressing atypical B cells, which are shown in green. CD44 high, CD11c negative B cells, which we think to be germinal center derived memory in blue. And then some earlier phenotypes of cells are expressing Ki67 or expressing CD62L. We used pie charts in F to describe the relative frequency of each cluster at each time point. You can see at day 7, early in the response, there is a wide mix of clusters, and as the germinal center becomes predominant, the red cluster becomes by far the largest. And at a later time point, we then see a re-expansion of other clusters, namely the CD44 high, CD11c negative cells, which we believe to be germinal center-derived memory. In G and H, we quantify the relative frequency of each cluster and the total number of each cluster, or total number of each number of cell in each cluster based off the total size of the lymph node. For supplementary figure 2, we performed similar analysis, but this time looking in the spleens of the mice. This is because on performing intramuscular immunization, the spleen acts as a secondary draining site, and so we find IgD-negative RBD binding B cells in the ChAdOx1 nCoV-19 immunized mice in their spleens, but we do not find such cells in the ChAdOx1 over-immunized mice, shown in B, again in the blue and green lines. And we performed similar t-SNE and quantification analysis, finding all of the same clusters and all of the same conventionally gated cells, with the addition of one extra cluster shown here in grey. These are the Ki-67 negative CD62L negative cells, which we presume to be some form of early progenitor, which predate the germinal center formation. And then we have the quantification here in G and H. So largely the spleen recapitulates the data that we saw for the lymph node. In figure 2, we introduce a mouse model. This is the CD4 BCL6 flox mouse model. And so here, any cell which expresses CD4 will express the Cre protein and will therefore flox out BCL6. And this essentially is a Tfh depleted or Tfh deficient mouse. Tfh are T follicular helper cells, of course, and here we define them as PD1 and CXCR5 double expressing cells. In the mouse that has the inactive Cre on the left, you can see this population of PD1 and CXCR5 double positive cells, and this population is lost when the Cre is active. The quantification is shown here in B at days 14 and days 42 post-vaccination for the two groups of mice, and you can see that when the Cre is active, we have a complete loss of T follicular helper cells. Then in C, we looked at the germinal center here on the left, BCL6, Ki-67 expressing B cells, and within this population, we looked at the RBD binding B cells. And here on the bottom are the CD4 Cre animals. What you can see is that the germinal center B cell compartment is heavily reduced, but importantly, there is still a small number of GC B cells left surviving. However, when we look at these cells, their ability to bind RBD is not present. And this means that we have lost the ability to select the RBD-specific B cells and give them the necessary signals needed to proliferate them out and expand them beyond the realms of a normal clone. This is quantified out in D. You can see that the green bar, the green dots are essentially on the x-axis. There are very, very, very few RBD-specific germinal center B cells found in these mice. And what this tells us is that Tfh cells are required for the efficient generation of RBD-specific germinal center B cells. In the second half of the figure, we did some serum analysis from these mice. So we took serum at day 42, and we looked for RBD or spike-specific IgG in the serum of these mice. And what you can see is that mice with CD4 Cre BCL6 flox active have a greatly reduced amount of IgG, both for anti-RBD and for anti-spike. In F, we did isotype analysis, and we can see that there are differences in the amount of IgG1 and IgG2b. Namely, the IgG1 is reduced when CD4 Cre is active. And this goes with what we know in that the germinal center effectively supports the production of IgG1 specific germinal center B cells. Finally, for figure 2, we used a pseudo-neutralization assay, and we looked for IC80, which we quantified here. You can see a large reduction, essentially to the limit of the assay's detection, in neutralization capacity of the serum when CD4 BCL6 flux is active. So when Tfh are lost, not only do we lose RBD-specific germinal center B cells, but serum immunity is heavily depleted. Moving on into figure 3, we continue to look at these mice, now looking at IgD negative RBD-specific B cells in A, and we quantified out the number of IgD negative and IgD positive RBD-specific B cells in the lymph node at day 14 and at day 21. You can see that loss of Tfh results in a reduction in the number of IgD negative RBD-specific B cells, the stats for which are shown in green in figure B. We then took the subpopulation gating used in the t-SNE analysis of figures 1 and supplementary 2, and we applied it to these mice. And so we described the various subpopulations of IgD negative RBD specific B cells. We have the CD44 expressing cells in blue, the CD11c expressing cells in green, and so on. And essentially, all of these populations are reduced in the mice which have CD4 active with BCL6 being floxed. And so this tells us that Tfh cells are required for all of these RBD-specific subsets. The only one that is slightly different here is that at day 42, the CD11c isn't quite as significant, suggesting that these atypical cells may have some route beyond that of the germinal center in order to be derived. In supplementary figure 3, we performed similar analysis again, this time on the spleens of these mice, and we come to the conclusion that the spleen has some Tfh-independent subsets of IgD-negative RBD-specific B cells, namely these memory cells in blue and the CD11c cells in green. So this suggests that there are slightly different dynamics at play in the lymph node and in the spleen, but that the lymph node really is heavily Tfh dependent for generating its RBD-specific, antigen-specific B cell subsets after an immunization or vaccination. In the second half of figure 3, we introduce a second mouse. This is the S1PR2 ERT2 Cree Rosa 26 stop flox RFP mouse. So in this mouse, we have S1PR2 having this ERT2 Cree motif, such that when the mouse is treated with tamoxifen, it will produce the Cree protein in any cells expressing S1PR2 at the time. And then the Cree will allow this stop flox RFP motif to become active on the Rosa 26 locus, which is very widely expressed. And what this essentially will mean is that at the time of tamoxifen administration, any cell expressing S1PR2 will become labeled red using the RFP. And so we immunized the mouse, waited until days 8 and 10, at which point we gave tamoxifen, and then we looked at day 14. And the concept here is that we are going to label germinal center B cells and also label germinal center Tfh. Both of these populations are known to express S1PR2. And so the quantification is shown in E. You can see that by flow cytometry, you can clearly detect a population of RFP positive cells. These are cells which are being reported for being positive for S1PR2 expression. And we can see that on the Tfh, which is the pre-gating shown in E, so CXCR5 PD1 double positive, CD44 positive, and CD62L negative, we have a healthy population of S1PR2 labeled Tfh. We then compared the RFP positive to RFP negative Tfh, and we show that the cells which are labeled with RFP therefore have S1PR2 expression, have a higher expression of CXCR5 and higher expression of PD1. And this goes with the idea that S1PR2 is a marker of terminally differentiated, highly active germinal center Tfh. We performed imaging using a Stellaris 8 Leica microscope on whole lymph nodes, and here we were able to image the S1PR2 again, this for RFP shown in red, and we included 4 other markers in our confocal microscopy. We included CD3 in green, CD35 in white, Ki67 in dark blue, and IgD in cyan. So CD3 marks the T cells, and we can see in the center of the lymph node the T cell zones. CD35 marks follicular dendritic cells, and this can be seen in the germinal centers. Ki67 marks proliferating cells. Of course, B cells proliferate largely when they are in germinal centers. And IgD is marking the juvenile naive cells, the B cells which are not germinal center B cells but are found on the outside of these structures. So on the left we have S1PR2 being active. We can see the red staining, and on the right we have a control mouse in which there is no red visible under the microscope. So S1PR2 is not active, or the S1PR2 RFP labeling is not active. In H we have a zoom, and here you can see a follicle which is boxed around with a white box, and this is then further zoomed in I. And what we can see is S1PR2 overlaps both with CD3 positive green T cells, but also with other cells which we know to be the B cells. And so we can say that here we have germinal center Tfh and germinal center B cells being generated by the Chaddox vaccine, and this is some nice imaging which we were able to include. Supplementary figure 4 has more lymph nodes available, and this is just to show that our data is quite reproducible, and in various lymph nodes, both for mice with S1PR2, ERT2 Rosa 26 stop flux RFP being active, we can see the red cells within our germinal centers, within our follicles, and in our control mice there is no red staining, showing that this mouse works really quite nicely. In figure 4, we include some human data in the paper, and this is from a collaboration with Dr. James Thaventhiran, also in Cambridge, and he had access to a patient who had a CD40 ligand mutation such that we can consider them to almost be Tfh deficient. And so in this patient, we see a lack of IgD and IgM double negative mature class switched B cells, and within this pool, we see very, very few RBD binding B cells. And we can compare these both to people vaccinated at the same time, but with an irrelevant vaccine. So this is the meningitis vaccine shown here. And for patients receiving their COVID-19 vaccines in the bottom row here, in which case we can pick out RBD-specific B cells. And so we quantify the number of RBD, or the frequency of IgD negative RBD specific B cells, here in black for the healthy normal individuals, and in red for the patient with a CD40 ligand mutation. We performed the pseudo-neutralization assay in C, and we did anti-RBD IgG ELISA in D. And what we can see here is that this data correlates with our mouse data, showing that CD40 ligand signaling, which can be taken as a proxy for Tfh activity, is required for circulating RBD binding B cells to be generated and for serum immunity after human vaccination. Moving on, we then went to look at a mouse that had germinal center B cell deficiency. And we set this up using a mixed bone marrow chimera system, as shown in A. So we used RAG2 recipients, and we gave them either CD23 wild-type BCL6 flox bone marrow, this is essentially wild-type bone marrow, or CD23 Cre BCL6 flocks bone marrow. So CD23 is expressed on all mature B cells, and so these cells will have no BCL6 within their mature B cells, and this will render them unable to generate germinal center B cells. So we performed the irradiation and bone marrow infusion. We allowed 8 weeks for reconstitution. Then we vaccinated with ChAdOx1 and COVID-19, and we looked at 2 downstream time points as previous. In B, we gated our germinal center B cells. This time we used Ki67 positive and CD38 negative B cells as our germinal center gate. And we showed that the mice which received CD23 Cre BCL6 floxed bone marrow had a deficiency in germinal center B cells. This is quantified in C. Germinal center B cells and T follicular helper cells have a somewhat symbiotic relationship, and so we were not surprised to see here that the CD23 Cre receiving mice had a deficiency in their T follicular helper cells as well, again described as PD1 CXCR5 double expressing CD4 T cells. And the quantification for this is shown in E. We performed more confocal imaging. On the left is a mouse which is essentially a wild-type mouse, and you can see these clear follicles of Ki67 expressing B cells surrounded by IgD negative cells on one side and the T cell zone on the other side. And within the follicle we can see some white staining correlating to the follicular dendritic cell network. However, in the mice which had CD23 Cre active with BCL6 being floxed out, we had a loss of structure. There are now no clear follicles which contain follicular dendritic cells and proliferating IgD negative B cells. And so unsurprisingly, when we looked in the serum, we saw a strong decrease in anti-spike and in anti-RBD IgG, and a decrease in neutralization capacity of the serum. And so quite unsurprisingly, germinal center B cells are required for effective serum immunity following ChAdOx1 and COVID-19 vaccination. Supplementary figure 5 comes next, and this includes more lymph nodes which you can look at, essentially just proving that our staining is quite reproducible. So then in the second half of figure 6, we return to our S1PR2 labeling system, and this time we looked at the S1PR2 labeled B cells. And around 10% of B cells were S1PR2 RFP labeled at 14 days post-vaccination. We looked at the RFP positive and the RFP negative subpopulations for RBD binding, and we found that the majority of RBD binding B cells are found in the RFP positive population, despite this population being much smaller than the RFP negative population. This is quantified out on the right hand side here. Then taking the S1PR2 labeled RBD-specific B cells, we looked at our various subpopulations of cells. This time we gated germinal center B cells as GL7 high and CD38 low cells, and then we used our familiar gates of CD44 and CD11c, Ki67 and CD62L to describe the various subsets at day 14, which we show in G in the pie chart. Finally, in figure 7, we introduced a prime boost experiment into our study. So we used the S1PR2 labeled system. We immunize, give tamoxifen, and look at day 14, as we did previously. But then we also had a subset of mice which we left for a longer period of time before giving a second vaccination on day 42, and then we analyzed the recall response 7 days after this on day 49. And at day 49, we could still see S1PR2 labeled B cells. Now remember that these were cells labeled at days 8 and 10 when tamoxifen was initially given, and so these are cells which were generated in the primary response and have lived up until this point and have now been recalled. If we look at the germinal center, described by GL7 positive CD38 negative B cells, we can see that at day 14, almost all of the GC is S1PR2 RFP labeled in red. At day 49, these cells are now less frequent. However, there is still a significant minority of cells in the germinal center which are RFP labeled. If we flip this around and we ask what is the phenotype of our RFP labeled cells at each time point, we get the same answer, and that is that the majority of RFP labeled cells are germinal center. They are GL7 high and CD38 low, both in the prime response when we gave the vaccine, then labeled and then looked, and in the recall when we waited for an extended period of time, then gave a second vaccine and then looked at the recalled cells. So this is suggesting that when cells are recalled, primarily they are recalled as germinal center B cells. This is then quantified out in ENF using our gating as previous, looking at day 49 RBD binding RFP positive B cells. F shows the stacked bar charts in which you can see that at day 49, around half of the cells are germinal center derived B cells. And then we have a large number of CD44 high B cells, and so we think these are germinal center derived memory B cells which have been reactivated. In the second half of figure 7, we look at some other cell types, also recalled. In G, we look at plasma cells, which we describe as CD138 high and IRF4 high cells. And we can see that a large number of the plasma cells which are found in the secondary response at day 49 are indeed RFP labeled. So this suggests that their origin was from a germinal center B cell, which was S1PR2 labeled during the primary response, whose progeny were then recalled at day 49 as a RFP labeled plasma cell. In the remaining part of figure 7, we look at the CD4 T cells again, and we can see that we find RFP labeled T conventional cells both at day 14 and at day 49. If we look at the phenotype of these cells, what we see is a high frequency of CD44 expressing cells, so these are the activated non-naive cells. And within this population, we see PD1 and CXCR5 double positive Tfh, shown in blue. And in the non-Tfh cells, we see Tbet and CXCR3 double expressing cells, which are Th1 cells. So at day 14, the predominant cell type is Tfh in blue. At day 49, and this is during the recall response, we do still see a large number of Tfh. However, we see a relative expansion of the number of Th1 cells, and so this argues that Tfh generated in a primary immunization can be recalled once again either as Tfh or as Th1 cells. So that's the data of the paper. I'll conclude by highlighting the graphical abstract which we include, as well as our key messages. And these are that Tfh cells are required for RBD-specific B cell selection after Chalux-1 and COVID-19 vaccination, that an absence of germinal center B cells severely limits the capacity of the mouse to produce RBD-specific B cells and limits serum immunity following Chalux-1 and COVID-19 vaccination. We show that RBD-specific B cells are recalled to the germinal center during secondary responses. We did this using our S1PR2 labeling system. And we showed that GC Tfh cells are recalled during primary responses either again as Tfh or they can be recalled as Th1. So finally, I will thank the BBSRC and the Lister Institute for funding my work. I work at the Babraham Institute, and you can contact me at my email or on my Twitter or on my LinkedIn if you have any questions about the paper or if you'd like to know more. Thank you.